Hi everyone, welcome back to Useful Genetics. This is Lecture 2C, where we're going to talk about why most mutations are harmless. And there's really 2 points. Most new mutations are not in genes, and even when they're in genes, most of the mutations in genes are in introns. Now, here's a diagram based on analysis of the human genome. We could draw a similar genome for most other plants and animals because humans are really fairly typical. So the first point is most DNA is not part of genes. Half of our DNA is the kinds of sequences that are called genetic parasite sequences. They're repeated sequences we have many copies of, different categories called lines and signs and LTR transposons. We'll talk more about these later in the course. The main point now is that they don't do anything to serve the functioning of the organism. So another 20% of our DNA here is sequences with no known function that's not genetic parasites. There's also a chunk here, about another 10%, that's repeated sequences like the genetic parasites, but these sequences don't actually look to be parasitic sequences. They're just repeats that don't have any known function. So only about 28% of our genome is genes. I've superimposed a dartboard on the drawing of the genome to make the point that we can think of our genome as kind of a target for mutations. The mutations, however, are blind. It's as if the person throwing the darts was blindly throwing darts at the dartboard, and we expect they're going to land randomly all over the genome. So naturally, most of the mutations are going to land in most of the genome, which is the non-functional parts of the genome. This segment here is going to get most of the random mutations. Now, even when we consider the part of the genome that consists of genes, most of this DNA does not code for proteins. Only about 2% of our genome actually codes for proteins. The rest of this 28% is intron sequences. You remember that introns are segments of sequence that are cut out of the RNA molecule after it's been transcribed from the DNA, and only the remaining coding sequences become part of the messenger RNA that's actually translated into proteins. So introns are also largely non-functional DNA, and that's where the bulk of the mutations that land in genes happen. They just, by random chance, most of them are in introns. It won't take long now to describe what we've done. We've considered the reason that most mutations don't cause us any harm or have any effect at all is because most mutations are not in genes. They're in the very large fraction of our genome that's non-functional sequences. Even within our genes, most of the mutations that happen are in introns. Most of the sequences in introns are non-functional, and mutations there don't make any difference. It's only a small fraction of our genome that's coding, and even within that, there are differences in how much different sequences matter. Coming up next, Lecture 2D, we'll talk about mutations in coding sequences that don't affect function. We're going to reserve the discussion of functional consequences largely to Module 3. So here we'll be talking about neutral mutations and silent mutations. I hope to see you there.